Lorelle Sherman, and she is a forest ecologist, bird enthusiast, and wild foods forager. Foraging for mushrooms and bird watching for over 10 years inspired Lorelle to study the forest ecosystem she was spending her time in. Lorelle studied forestry and wildlife biology at the University of Vermont and forest ecology while at Oregon State University. Today, she serves as the OSU Extension Forester for Benton, Linn, and Polk Counties, where she hopes to inspire people to connect with and steward the land. So, Lorelle, thank you for joining us today.
Welcome, everybody. I'm here to talk to you about my passion outside of teaching forestry and forest ecology. I've been a mushroom enthusiast my entire life. Grew up in the Appalachian Mountains and picked morels with my family when I was very young, and moved to the Pacific Northwest and kind of fell down the rabbit hole of fungi that are here. So, today we're going to talk about fungi of the Willamette Valley, and we're going to go over what is a fungus, different types of fungal life strategies, including mycorrhizal, which is in the title, but we will also talk about wood decay and different types of fungus we're going to find in the Willamette Valley, including Marion County, the role that those fungi play in the forest, and then we'll go through some resources at the end. And we'll do questions at the end just because your question might be answered throughout the presentation. Alrighty, so what is a fungus? Fungi is just the plural of fungus, and you can pronounce either multiple ways, and they're all correct. Whichever way, fungi, whatever you want, they're all correct. But a fungus is a multicellular organism other than yeast, which is technically in the fungal kingdom and single cellular. They are distinguished from other kingdoms by a few different things. So they lack chlorophyll, and therefore they are not plants, even though they were included in the plant kingdom until I think 1969, which is very recent, shockingly recent. I think they were put in their own kingdom the same year that we landed on the moon. So there were scientific advancements enough to land us on the moon, but we still didn't know that fungi were not in the same kingdom as plants. But we do now. And they, oddly enough, are actually more similar to animals in the way that they get their nutrition. So they excrete enzymes to decompose complex molecules and then absorb those nutrients. And so that's a little bit interesting. But the other ways that they're different or unique, I should say, they're in terms of structure. Their cell walls are made of chitin, which is the same material that insect exoskeletons are made out of. So that is quite unique. And their growth strategy is through hyphae or mycelium, which you might have heard those terms, and we'll go over what those are next. And then reproduction. They have an incredibly complex reproductive system, numerous different strategies. I'll touch on it, but I won't go into it because that would be an entire other webinar. So when we're talking about what the building blocks of a fungus are, if we see in the top right picture, there are these little rectangles. Those are the cells. And the white lines, those are the cell walls. Remember that they're made of chitin, which is that exoskeleton insect material. And they form these long strands called hyphae. Hyphae is the building block of a fungus. And the hyphae kind of they grow into each other, overlap each other, they fuse, and they create these fungal mats, or these mycelial mats, which you see in the bottom right. So mycelium is essentially just a big mass of hyphae. And the growth occurs at the tips of the hyphae, and they'll continue to just grow into each other and create this dense mat. So when we see a mushroom, like on the top right, that is just the reproductive structure of a fungus. So the main organism is actually that mycelial mat, which you see is underneath the soil or in the soil. And when the environmental conditions are correct to cue fungal fruiting, the fungus creates a mushroom fruiting body from that hyphal mass. Each species of fungi has a different environmental cue. They often have to do with some combination of soil temperature and moisture. So, like if you're going morel hunting in the spring, a lot of folks who are really into it take soil thermometer or just a thermometer out and stick it in the soil and see what the temperature of the soil is, because they won't fruit under a certain temperature. It's important to note that not all fungi produce mushrooms, and we'll talk about that a little bit more. But so just note, when you see the mushroom fruiting body, that is not the entire organism. That is really just the reproductive structure. Some people relate it to an apple on an apple tree. That's kind of a good way to look at it. So I mentioned that the reproductive strategy is complex, but what we can say is that they produce a lot of spores. This can happen asexually or sexually, and they have many different dispersal methods. Most are wind dispersed, as you can see in that left photo of a puffball, a lycoperdon. But there are other methods. So mechanical, some get stuck to insects and animals, and then there's a unique water example that I will show you, a rain specifically next, and we'll talk about that in the next slide. But yeah, it's important to note the mushroom spore shape, size, and color when you're trying to identify a species. That can be integral for species identification. A lot of mushrooms look identical, except when you look at the spores. So one way to check out the spores is by doing a spore print. And on the right, you can see that Agrosity actually did its own spore print, so you wouldn't have to, because it dropped its spores on the mushroom below. So we know that those spores are chocolate brown, and you could scrape off a little sample, put it on a microscope plate, and look at what the shape is and the size and the color. But you could cut the cap off yourself, bring it home, put it on a piece of paper, and cover it with a Tupperware to keep the moisture in, and let it drop its spores, and then look at it. The spore, it's really fun. You can get a cheap microscope. It's really fun to look at the spores. There's even a mushroom that has a penguin-shaped spore. You can look it up. There's pictures of it. It's pretty cute. So mentioning the spore dispersal methods, so there's this one really fun one, which is very common in Marion County and in the Willamette Valley. I'm guessing many of you have seen these when you're walking around in the woods. This is called a bird's nest fungus, and it's Nigella is the genus. There's several species. They're kind of hard to tell apart. But so on the top left, you see there's immature and then mature cups. The immature cups have a covering on them and to protect the spores while they're maturing. And then on the right, you see what looks like little bird eggs are actually packets full of densely packed full of spores. And the way that these get dispersed is through rain, and we have a lot of that in the Pacific Northwest, so it makes sense why we have a lot of these bird's nest fungi. So I found this diagram on the bottom. The citation is cut off, but I did not draw this. So on the far left, you see a raindrop hitting the cup, and it literally launches these spore packets onto the nearest surface, whether that's a plant or the ground. And so that's an alternate method, different from wind dispersal, kind of fun. And if you Google bird's nest fungi raindrop, there is a really spectacular video of this happening in action on PBS. So be sure to look that up. Maybe we can include that in the email that we send out. Okay, so we know what a fungus is. Now we're going to talk about the different fungal life strategies. So mycorrhizal fungi get most of the press, the attention, and we're going to talk about that for sure. But there's all these other life strategies that are integral for forest ecosystem health in the Willamette Valley and beyond. But we'll start with the mycorrhizal fungi. So, as you may have heard, the mycorrhizal fungi form a symbiotic relationship between the fungus and the root of a plant, whether that's a tree or an herbaceous species. And that means that both the symbiotic relationship is a net positive, so both species benefit from this relationship. In this relationship, the fungus gets carbon because remember, the fungus cannot photosynthesize. It does not have chlorophyll. So the plant is producing excess carbon and sugars and giving it to the fungus in exchange for the fungus giving the plant increased capacity to access water and nutrients. So mycorrhizal fungi are able to absorb and transfer all of the 15 major macro and micronutrients necessary for plant growth. And they do this because they can release powerful chemicals into the soil that dissolve hard to capture nutrients and break them down and make them into a usable form for plants and then give them to the plants. And so this extraction process is really important, in particular in places that are nutrient limited, poor sites. And it also kind of explains why non-mycorrhizal plants, which there aren't many, most plants do form some sort of mycorrhizal association, especially in the Pacific Northwest. Why plants that don't require high levels of fertility in the soil to maintain their health. So if a plant is not going to form a mycorrhizal connection, it better be living in soil that has plentiful nutrients and water that are in a form that is accessible to that plant. So on the right, you can see a little diagram of a mycorrhizal fungus that's connected to two different tree species. We'll get a bit more into this. And yeah, it's, all the mycelial network underneath the trees is where the magic is happening, and that little mushroom is just the reproductive system. So you might hear, mycorrhizal fungus in the news and whatnot, but it's important to know that there are actually 4 different kinds of mycorrhizal connections. The first 2 are quite common in Western Oregon and the Willamette Valley, so ecto and endomycorrhiza. And on the right, it's a diagram of what a plant root looks like with both of those. So ectomycorrhiza, if you look at that blue on the right to that diagram, the blue is supposed to be mycelium. It basically forms a protective mantle or sheath, like a sock almost, around the plant root. And on the right, so it says it's labeled arbuscular. Endomycorrhiza and arbuscular are interchangeable, and folks actually more commonly say arbuscular now, but we used to call them endomycorrhizal, so it can be a little confusing. They actually don't form a protective sheath on the plant root. They instead penetrate the cell walls. And then orchid mycorrhiza, we clearly don't have those in Marion County, so I won't talk about them, but it's fascinating the unique connections between the root corms and fungi that have evolved there. We do have some ericoid mycorrhiza, and if you are a plant nerd, you might know that means ericoids are heath, blueberries, rhododendrons, manzanita, madrone, so certainly things that we have in the Willamette Valley. These mycorrhizal connections actually play a crucial role in nutrient acquisition in nutrient poor and acidic soils. And so if you've ever grown blueberries, you know that if there's acidic soil there, or they do well in acidic soil. And so yeah, that all plays into these different connections. So the type of mycorrhizal fungi a plant partners with has really big implications we've recently discovered. So evidence is now showing that the dominant fungi of a region can actually influence the overall health and diversity of forest ecosystems. So in the Pacific Northwest, we have mostly the ectomycorrhizal connection, so that blue kind of a sock over the root systems. And researchers have found that ectomycorrhizal fungi actually, through studies, they showed that the trees and plants that were associated with these experienced positive feedback in biomass. So they grew bigger, denser, bigger diameter trees, taller trees, whereas plants associated with the endomycorrhizal or arbuscular actually had negative feedbacks in biomass, which is very interesting. They repeated these experiments with seedlings and also found that seedling survival was reduced for arbuscular or endomycorrhizal associations, whereas seedling survival increased in ectomycorrhizal. So why might that be? This is the last sciencey diagram, I think, that we can get through. So on the right here, so we notice there are these major differences between the 2 strategies of ecto and endomycorrhizal fungi and how they attach to the plant root. This is a cross section of a plant root. So it seems to come down to the way in which they form root associations. So on the right, there's that ectomycorrhizal fungi that forms that protective sheath around the roots. It's like a tight stalk of dense filaments, and it actually acts as a physical barrier against the invasion of root disease. And not only that, they excrete the mycorrhizal, the ectomycorrhizal fungal sock, we'll call it, excretes antibiotics to immobilize and kill disease organisms and pathogens in the root zone. So they haven't even gotten to the plant root yet. So that's pretty impressive. Whereas on the left, it's showing that arbuscular or endomycorrhizal strategy, and they penetrate the cells and do not offer the same level of pathogen suppression to the plant. So it's kind of interesting. On face value, this seems like it's bad news to have an endomycorrhizal connection. But if you look at the grand scheme of things, the negative feedback experienced by these endomycorrhizal fungal trees may lead to more diverse forests because they're suppressing any one species from taking over, giving many more species a chance to grow up and get big and tall. If we're looking at the Willamette Valley, though, we don't really have a huge amount of tree diversity. Like think of all the Doug fir we have compared to tropical regions or just other farther south, I guess. And so that makes sense because our trees are almost exclusively ectomycorrhizal. So that's to me just really fascinating that these fungal connections are actually it's changing how our forest ecosystems look above ground as well. And these connections have formed over very long periods of time. So just be aware that when people say mycorrhizal fungi, that isn't they're not all the same. They do very different things. All right. So when we're talking about ectomycorrhizal fungi, which is what we have most of in Oregon, um, we are talking about, so there's differences between specificity. And what I mean by that is, I'll use a couple examples to explain it. So here I've got this kind of cartoon drawing of a Douglas fir. Douglas fir is a generalist ectomycorrhizal host. And so that means it loves mushroom. It just loves connecting to mushrooms. And it's kind of willing to connect to a lot of different species. So that's called a generalist behavior. And so we've got, it connects to black truffles, um, other non-culinary truffles, chanterelles, boletes. This is a Xericomellis bolete, swillus, which is slippery jacks. They're really slimy. Amanita muscaria, kind of just so many different types of fungi. However, each of these fungi have a different specificity themselves. So the Doug fir is willing to connect to all different types of mushrooms or fungal species, and then we'll go through each of these. So this black truffle only grows on Doug fir. So it's not an even relationship between the specificity of the Doug fir and the black truffle. This rhizopogon, only Doug fir, maybe one other conifer species. Pacific chanterelles, maybe two to three species that the chanterelles will grow on. Xericomellis, they grow on several conifers. Swillus grow on a bunch of different conifers, and Amanita muscaria literally grow on almost anything. So you can see that while the Doug fir itself, it has a generalist host specificity. That doesn't mean that the fungi it's associated with also have that generalist specificity. So this Amanita muscaria, like I said, it literally grows on just almost anything, even non-native species. Big leaf maple, oaks, birches, spruces, hemlocks, Doug fir. I mean, I'm sure you've seen it pop up all over the place. I'm on Oregon State University campus, and it grows in the medians between the parking lots. It's just kind of everywhere. So you can imagine that this Amanita muscaria in certain situations might actually be connecting the mycelium or connecting two different tree species through its mycelium, which is interesting. The data on how that plays out we're still figuring that one out. How different species of trees interact with each other through the mycorrhizal network of generalist mushroom species like this Amanita muscaria. On the other hand, we have Oregon white oak, which has more of a host fungi specificity. So The oak, this is a black trumpet mushroom. It only grows on Oregon white oak in the Willamette Valley because we don't have ginkgopin really in the valley. But so it's a 1-to-1. The Oregon white oak associates with black trumpet and black trumpet with white oak. And then there's a couple of others that only associate with white oak. So this is a non-culinary truffle called a genia. Pistophilis tinctorius, which is a dye mushroom, and they look like just kind of, they're called dead man's feet when they come up. They look like big balls, but if you cut them open, they're gorgeous inside. And then here's another truffle. So oaks actually associate with a lot of underground truffles, and that's because they grow in these really harsh habitats that get sun-blasted in the summer. And so a lot of their mycorrhizal fungal associates are underground to keep from totally drying out in the summer. Alrighty, so I wanted to spend that time talking about mycorrhizal, but leave enough time to get into what these other life strategies are because they are super important for our fungal or for our for our forests. So saprophytes. Saprophyte is a fancy word for a wood decay fungus. So wood decay fungi decompose wood. They break down organic matter and they make stuff decompose. And without, they're really good at what they do. They saprophytic fungi can break down dead plant material, but also animal material. And without them, we'd literally be overrun with rotting, decaying, dead matter, just logs all over the place. So we really need these saprophytes in our forests. When they're decomposing wood, they actually make the carbon and other nutrients. They turn it into a usable form for plants. So if you see a big down log on the ground, plants can't access the carbon and nutrients in that. But once they break it down, the fungi are releasing those nutrients back into the soil for future plants. And the identification of saprophytes is grouped by the method of degrading wood, which you can see on the bottom right. There's all different types of resulting wood decay. There's different wood decay pathways. And then also whether or not they're a primary, secondary, or tertiary colonizer. Primary being they can actually sometimes lean into being parasites, which we'll talk about. And that means they can sometimes attack trees that are alive and well, or trees that are stressed, a little bit stressed. Whereas secondary, they rely on primary colonizers to break down wood in the tree, and they can't attack a live, healthy tree. They're going after standing dead wood, whereas a tertiary colonizer is going to be the stuff that's finishing off those logs on the ground. So I have some photos of that too. But yeah, just remembering that between mycorrhizal connections and the saprophytic fungi, otherwise known as wood decay, mushrooms are at both sides of the carbon cycle. So they are storing carbon through the mycelial mat of the ectomycorrhizal fungi, or endo, and they're releasing carbon by breaking down wood. So they're on both sides of that carbon cycle, which I think is quite interesting. So in the Willamette Valley, in our forest ecosystem, so if you're walking around in your woods, there are two major types of wood decay fungi or saprophytes. The first is white rot. The second is brown rot. So white rot is more aggressive. Usually these species are more aggressive. So think primary colonizers, not secondary or tertiary. And they break down pretty much all of the wood components, cellulose, hemicellulose, and lignin. And on the top right, so these photos on the right are both honey fungus, otherwise known armillaria or honey fungus. If you've heard of the humongous fungus, that's an armillaria. It's I think like the biggest mushroom in the world. I'd have to fact check myself, but so on the top right, you're seeing the mycelium of that honey mushroom kind of eating away at the wood of this Doug fir tree. So this is actually leaning into parasitism, but it's breaking down the wood, so it's also saprophytic. Bottom right is the fruiting body of the honey mushroom, which is edible for some people. For most people, some people have issues with eating it. The other group is brown rot, and these tend to be those secondary and tertiary colonizers. They are less aggressive, and less often lean into that parasitic realm. And you can, so the white rot degrades cellulose, hemicellulose, and lignin. Brown rot does not. It's not able to degrade lignin, and lignin is blocky in nature, so it results in these big blocky stumps or logs. And I know I see that all the time when I'm walking in the forest, the big blocky stumps. So there's a lot of species in this group that are edible and medicinal, and I've called out two of them. So the bottom left two photos, that is chicken of the woods, Latoporus. It certainly can be found in the Willamette Valley, and it is a delicious edible for many. Some people have gastro problems with it. Some people have gastro problems with any mushroom, really. But and then this one, I think it's funny when you're a mushroom hunter and you want to eat the mushroom, it's called chicken of the woods. But if you're a forester, this is called dark brown cubicle rot mushroom. So I think it's kind of funny to look at the different perspectives of mushroom names. So on the right, there's two photos. And this one is Fomitopsis panicola, incredibly common in the Willamette Valley. And this forester's name, the common name is red-belted conch, and the forester's name is brown crumbly rot mushroom. So I mentioned that parasite life strategy where some of the saprophytes kind of lean into that. Parasitic fungi are causing harm to the organism that they're feeding off of, and that they're usually going after stressed trees. There's only a very small handful, maybe just one or two in the Willamette Valley that can go after a totally healthy tree. But some of the common ones that we see in the Willamette Valley that waft or they flip between saprophytic and parasitic is cauliflower mushroom, which is this one that just popped up. Honey mushroom was a couple of slides ago. And then Dyer's polypore, which is shown in this background photo here. And this one you'll see all over the woods in Western Oregon. It feeds off of the roots of conifers. So when you see it in this photo, it's actually kind of degrading the roots of this tree, and it's a sign that you might consider cutting this tree down. It might become a risk in the future. One fun example of parasitic fungi that I would imagine a lot of us have seen is the lobster mushroom. This is a common mushroom in Western Oregon in the Willamette Valley, and it is actually a fungi on fungi parasitism. So in the far left photo, you see a kind of just like your standard white mushroom with gills on the right. And then on the left, a very odd orange looking mushroom. And so on the right, that white mushroom is called a Russula brevipes. It is edible, but it's very bland tasting. And what happens is this fungal mold called Hypomyces lactiflorum specifically attacks that Russula brevipes and alters its DNA, alters the DNA and metabolites. So it's completely changing the mushroom into a different species, essentially. And you can see in the center, there's some photos of the progress of that. Starting at the top, there's a intact Russula brevipes, and then it's starting to get parasitized, starting to get that orange color, and then at the bottom, it's fully parasitized. So there is little to no DNA in that orange lobster mushroom that looks anything like that white Russula brevipes, which is really shocking to learn. And the Hypomyces lactiflorum actually alters the metabolites as well, and that changes the taste and edibility of the mushroom. So the initial Russula brevipes is bland, and then the resulting lobster mushroom is much more flavorful and delicious. So people don't tend to harvest that white Russula, and they just look for the lobster mushrooms. And then I'll real quickly go through these last two because for the sake of time, essentially. Endophytes, they live intercellular and intracellular of essentially all plants on Earth. And they're beneficial to both, it's a symbiotic relationship. They're incredibly important for plants, and we're really just starting to scrape the surface about these. They exist in the cells of plant tissue and just live there and for years and years and years, and we just started to detect them. They're actually producing secondary metabolites that are antifungal, antibacterial, antimicrobial, that are protecting the plants. They activate the plant defense system. It's really remarkable, but I'm not going to spend much more time on it here. And then lichen, it's just important to remember that lichen is a hybrid colony of fungi and algae or cyanobacteria. So in the photo on the right, you see an algal, kind of a ball of algae, and that's the photosynthesizing plant, essentially. And then it's being held by the hyphae of a fungi. There is also yeast that comes into play there. We just found that out in 2016. But something that I wanted to bring up with lichen, being in the Willamette Valley, we often see big leaf maple and other hardwoods, but mostly big leaf maple, being totally covered in lichen and moss. And I often get asked if that's harmful for the tree and if it should be removed. There's even one tree that in the study that I'm about to talk about had 78 pounds of moss and lichen on one individual big leaf maple. But what we've recently learned is that the big leaf maple and vine maple actually tap into, so I'll back up. So those moss and lichens that are on the tree are trapping dust particles and soil particles in the air, and they're creating canopy soils up in the branches of the tree. And the big leaf maple is actually creating canopy roots into those canopy soils and tapping into the lichen and moss, water, and nutrient content. So this is important for in the Willamette Valley. You know, we've been having lots of droughty summers, high temperature, high heat summers. And these mosses and lichens hold on to moisture and nutrients for a lot longer than the tree can itself. And so not only is it utilizing ecto and endomycorrhizal connections in the soil, on the ground under the earth, it's also putting roots out into these canopy soils. So just really wild. There's some fun studies on that from, I think, Washington State, if you want to look that up. And so just real quick here at the end, I know we're approaching the end point here. We started a bit late, so I'll try to go real quick through this. There are other ways that fungi are important in the forest. We already went through mutualisms, nutrient recycling, which is the saprophytes, wood decay fungi, pathogens. So the one thing I wanted to touch on is how fungi are important for reforestation and wildlife, food, and habitat. So the timber industry has been inoculating their seedlings prior to planting since the 90s. And they're inoculating with, because we're in the Pacific Northwest, ectomycorrhizal fungi. In other places, they are trying to do endomycorrhizal, but here mostly ecto. And the community of fungi in wild soils can be complex and incredibly hard to mimic. So sometimes they actually go out and collect soil or collect fruiting bodies and create like a slurry. So they're utilizing what's native on the site. It has to be very site specific, very localized in order for it to work. So when you go to Home Depot and buy a bag of mycorrhizal fungi, whether or not that's going to work in your forest is up in the air, essentially. And then this is just showing from a study where they locally harvested ectomycorrhizal fungi and then inoculated tree roots. You can see the photo on the left, the tree on the right, the root system was inoculated with mycorrhizal fungi, whereas the one on the left was not. No question there about improved root growth because they had better access to water and nutrients through that mycorrhizal network. It's also important for fungi are also important for reforestation after fire. So before a wildfire comes through, you know, there's a mix of all these different life strategies that we talked about: mycorrhizal, saprophytic, endophytes. But post-fire, you think about all the dead wood on the ground. There's just so much dead wood that the fungal community largely switches to saprophytic. And if you've ever gone morel hunting in a burn, you'll see that because there's lots of cup fungus and just wood decay fungi out there. And they're breaking down all that big woody material and releasing the nutrients into the soil, kind of prepping it for future plants to grow in there, for future tree seedlings. And then areas of low or unburned soil that are nearby. You can see in the distance in this photo, there's some live trees. That's going to be your refugia for mycorrhizal fungi. And the fungal recolonization is going to happen from those areas back out into these burned areas, which have been prepped for tree seedlings by the wood decay fungi. So it really all works together. And then wildlife food and habitat. I do just have to mention this one because I am a wildlife person. And I actually studied how fungal decay pathways created habitat for my master's degree at Oregon State. You can see here on the left is a red-breasted sapsucker. That's a primary cavity nester. It's actually creating the cavity in the tree, but it relies on wood decay fungi to degrade the wood to a point where it can actually get in there. Almost all woodpeckers can't go after a totally healthy live tree. They require some sort of fungal decay pathway to start the process. And then on the right, you see a female purple martin nesting in the cavity and a big old conch on the right side of the tree there. And when I saw this, that's what started getting me curious about how wildlife habitat and fungi are connected, and they certainly are. Fungal decay pathways facilitate nest cavity excavation. I'm happy to send lots of information and research on that if people are into that. Oh, and there's just the male purple martin because it's beautiful. And then, so we'll end on this wildlife piece. Lots of species of fungi eat, or of wildlife eat fungi. You'll see the rodent teeth marks this is a spring porcini, but bears, ungulates, insects, soil microorganisms, they all love to eat fungi. And I think we will skip through this example so that we can get to some of the things that you might see popping up here in the Willamette Valley soon. So springtime is the time for morel mushrooms. You might have to go up into the mountains to get, well, let me back up. There are many different types of morel mushrooms, and they all grow in different habitat types. The one that's going to grow in Marion County right now is the one on the far left here. This is Morchella esculenta, and it's associated with riparian areas that have lots of cottonwoods and Oregon ash. So if you go out and walk the riverways, you might see this Morchella esculenta. That's a mycorrhizal mushroom. Whereas other types of morel mushrooms are saprophytes. So you can see there's one in an old community garden plot. You see all the squash seeds. That's a wood decay mushroom. So even within the Morchella family, there's all different types of life strategies of morels. And just to note, I just put this in here to make sure if you are going to go morel hunting that you know that there are lookalikes. This is a gyrometra, so just to be safe. And I know we're at 12:50. I still have more slides, but I'm wondering, I think we should leave time for questions as well. What do we think here?
It's up to you. You can keep going if you would like. I know we got started a little bit late, so you're welcome to
Okay,
keep going or
I'm going to skip just to the resources then. Real quick, we'll talk about this because I think it's important to leave you with resources.
we got a chat
So,
that says keep going.
okay, we will. So I'll present some resources that I use when I'm mushroom hunting. And when I say mushroom hunting, it certainly could mean for edible mushrooms, but I also do biodiversity surveys. And so it doesn't have to be for edible mushrooms. It could be for just for fun, seeing whatever types of mushrooms you could find. So I look at tree species maps. And why? Because remember we talked about those generalist versus specific relationships. If you have like a black trumpet mushroom that only grows on Oregon white oak, you need to know where white oak are. So I look at tree species maps, topographic maps, bird maps. So I'll just go through each of these. So this is a resource that's USDA Tree Map Explorer, and it's very landscape scale, it's coarse scale. You can search out, so you see on the bottom right, all these different tree species and see where they grow. It's a starting point. I would say use this to figure out what area to look in, and then you're going to need to use other tools. This is a topographic map, and I look at these all day long because a lot of the edible mushrooms that we like to eat like to grow in flat areas surrounded by steep areas. And that's because if you think about all the spring melting snow and landslides, all kinds of disturbance. They're bringing down all of this nutrient and water content to these lowlands, and a lot of mushrooms are actually disturbance fruiters. So what I do is I look at a topographic map where the lines are really far apart. That's a flat area or a flatter area. Where the lines are close together, it's steeper. And you can see that in this photo where I have circled where there might be lots of runoff into those areas. And so I could see big patches of disturbance fruiting mushrooms happening in those yellow circles. So that's where I would go. If you're looking for burn-associated mushrooms like burn morels, you can look at burn maps, and these are readily available online. I'm happy to include links in the email for this as well. I would recommend using a app such as Onyx Hunt or Gaia that tracks your walking path when you are not in cell service. So all you need to do is make sure your phone is fully charged and you possibly have an extra charger. And if you turn on your tracking, it will tell you exactly where you have gone, and it's quite hard to get lost. But then you can also mark where you've been and where you want to return to if you found something special. These also show land ownership, which is very important to know if you've crossed onto somebody's private land and you don't have permission to be there. iNaturalist, I shout it from the mountaintops every workshop I do, every seminar, because it's, in my mind, one of the most useful tools that we have. And it's helpful for identifying species, but it's also helpful for identifying where to look. So with the identifying species by photo, I know there are a lot of apps that do this, but I really do think iNaturalist is the best because they employ experts to go through and fact check their AI. So not only is it AI that's, it's getting better all the time for identification based on photos, but they have the leading mycologists and entomologists working for them to fact check that. So that makes me feel safer personally when using this app. And then if you're trying to find, so nobody posts their morel spots on here because they don't want you to go to it, but here's a workaround. If you know that the species of morel you're looking for associates with grand fir, you can look up grand fir and then go to those spots. So it's helpful for identifying where to look. Um, Yeah, and this is kind of what I was talking about. Some people did post their morel spots here, but not many, not too many. But you can at least get an idea of where to look. And then lastly, I will close out with unfortunate news is that there are AI-generated mushroom ID books, which are very incorrect. And they've been around for several years, but it wasn't until this past fall when a family actually used the book, and they ended up in the hospital. So my plea to you is look, these are all reputable books here. There's many others. When you're going to buy a mushroom book, Google the name of the author if you're buying it from the internet. And if nothing comes up, don't buy it. If you Googled any of these authors, lots of information would come up. So just trying to get the word out on that. Oh, and these are all AI generated photos too. So, we're in that era where we have to be aware. And that is all that I have.
But you mentioned a little bit about like inoculating plants with like a native mycorrhizal fungi, and it kind of made me wonder, are there any invasive species of mycorrhizal fungi that would be a concern or something to look out for, or can anyone do anything about that?
Yeah, there are, and it's an emerging issue, as you might expect. If you've heard of the death cap,
Mm.
which is an Amanita species that is deadly to humans and other animals. It's believed that it came over in soil in landscaping plants, but it's a non-native here and it's spreading. We don't see it too much in Oregon, but it's spreading in California, and that's a dangerous one. But in terms of ecological impacts, so a lot of there's big growing operations right now for edible mushrooms like oyster mushrooms, and oyster mushrooms in particular are very good at spreading and growing. And so there's concern that, especially the golden oysters, because they're being found in natural areas that we don't know the implications of that, but there's concern for what that might mean in the future. So yeah, so it's kind of an emerging issue at the moment.
Okay. And like some people, I think, will try to kind of like grow their own mushroom species in their yard, maybe. So is that something that could potentially be a risk of that spreading into the rest of their yard, or if they're next to a natural area, it could become an issue?
Yeah, I mean, there's plenty. So I grow shiitake and wine caps in my backyard. No risk there. Those are wood decay mushrooms that are real easy to grow, and I recommend trying it. But yeah, with the oyster mushrooms, um, I think the bigger concern, if you're next to a natural area, that you might want to avoid growing oyster mushrooms in your backyard. But if you're in an urban area, it's not, I don't know that it's too big of a deal there. I think I forgot to mention they're being used in restoration a lot. And so they're in the past, people have just been like, oh, oyster mushrooms, we're going to use whatever we have. And now we're realizing you have to actually use the native species. So I'm less concerned about people's backyard grows than these big, if you live next to a natural area and you're a huge growing operation, or if you're doing some sort of restoration project. Question in the chat. So I'm glad somebody asked because I feel like I should have added a bit more on that. So there at this time is no research that supports the idea that if you buy a bag of mycorrhizal inoculated soil from like a Home Depot or wherever, that it will work in your soil. And that's because, as we discussed earlier, or as I mentioned earlier, these are site conditions have these fungal communities are incredibly complex and site adapted. And you have these fungi in your backyard already. So if you buy a bag of inoculated soil and use it in your backyard, it is more likely to get outcompeted by native fungal mycelium, which is much more vigorous. However, you're mentioning potting soil for pots. It's really just I would love for it to say that it works, but we just don't have any evidence supporting it. However, like I mentioned with timber operations, they do see really positive results from using mycorrhizal fungi, but they're getting it from the site that they're going to plant the trees. So they're already using what they have there and growing it out. So it's a bit more complicated, I think, than going and buying a bag of soil from a store. Unfortunately, I wish, maybe someday research will come out showing otherwise.
All right. Well, it looks like that is all of our questions for today. So, Lorelle, thank you very much for joining us and giving your presentation. And thank you to Chelsea for helping out today and setting up the presentation.